The back of your eye is called the retina. When you see an object, light travels from the object to your eye. Photons enter your eye through the pupil and travel through the eye to your retina, where they encounter rod cells. Rhodopsin, a G protein coupled receptor in the membrane of the rod cells, is excited by the photon. Under dark conditions, rod cells work to inhibit bipolar cells, which serve as the middleman between the rod cells of the retina and ganglion cells that transmit information to the optic nerve and to the brain. But when it's excited by light, rod cells lose their inhibitory function and visual information is readily transmitted from the eye to the brain. But let's back up. In the plasma membrane of the rod cells, there is rhodopsin. Rhodopsin is coupled to a G protein, transducin, with alpha, beta, and gamma subunits. When rhodopsin is struck by a photon, it becomes excited. A component of the receptor, retinal, photoisomerizes from its 11 cis conformation to its all trans conformation. This induces a conformational change in transducin. The alpha subunit dissociates from GDP and binds GTP instead. It then dissociates from the beta and gamma subunits and activates cyclic GMP phosphodiesterase. The activated phosphodiesterase hydrolyzes cGMP into GMP, which reduces the overall concentration of cGMP in the cell. Decreased intracellular cGMP levels results in the closure of cGMP-dependent sodium channels, stopping the influx of sodium ions. The cell hyperpolarizes because potassium channels are still open and there is still an efflux of potassium ions. This hyperpolarization causes voltage-gated calcium channels to close, thus lowering intracellular concentration of calcium ions. Calcium ions are necessary for the release of the neurotransmitter glutamate. With a reduced concentration of calcium ions in the cell releases less glutamate, blocking the rod cell's inhibition of bipolar cells. The bipolar cells are now able to transmit visual stimuli to the ganglion cells, and it is relayed to the brain.